The overall goal of this procedure is to prepare electrocompetent and chemically competent E. coli cells for the introduction of exogenous DNA to measure the transformation efficiency and also to know the basis of the phenomenon of bacterial transformation to understand the difference between chemically competence and electroporation protocols.
When talking about introducing foreign DNA, it is important to differentiate between transforming and transfecting. The first one is exclusive for prokaryotes and the second one is referred to eukaryotes. The methods that can be used for eukaryotes are electroporation, sonoporation, asbestos tips, and chemical methods. When choosing a transformation method for bacteria, it must be taken into account the membrane composition, the size or the winding of genetic material, the time in which they were cultivated, and the material available in lab. The most used transformation methods are the ones that have been already mentioned: electroporation, chemically competence, sonoporation, physical treatment like asbestos tips, but there are also exist bioballistic, polyethylene glycol treatment, and ultrasound. The first one of these last ones inserts physically the DNA with a gold balloon with a shot. It has the disadvantage of the high cost. The second one is an organic solvent that acts at chemical level, but its efficiency is low. Ultrasound is versatile as it is less dependent on cell types than traditional methods. Of this method, the optimal in E. coli, it has been demonstrated to be at frequencies of 48 kHz for 10 seconds of exposure to ultrasound and after 24 hours of incubation.
It requires previous work of 2 days. The first day is shown by the stretch mark a strain of Escherichia coli in LV medium placed and incubated overnight at 37 Celsius degrees. On the second day, an isolated colony of top 10 strains in 5 ml of soft medium is grown in a test tube. This is incubated at 37 Celsius degrees with shaking overnight. The practice day, at least 3 hours before the start of this, 100 ml of LV medium are seeded in 1 ml of seed culture and incubated at 37 Celsius degrees as shown in Genetic Engineering Laboratory Manual. Once the pellet was obtained, supernatant was removed and resuspended in 10 ml of cold 0.1 molar calcium chloride and allowed to incubate on ice for 10 minutes. Centrifuge at 4000 rpm at 4 Celsius degrees during 10 minutes. Remove the supernatant and resuspend the bacterial pellet in 2 ml of 0.1 ml calcium chloride solution, 15% of glycerol. Incubate for 10 minutes on ice. 200 microliters aliquots are made and stored at minus 70 Celsius degrees, previously labeled microtubes. The following protocol was used for the measurement of transformation efficiency of chemically competent cells and electrocompetent cells. After having spatulated electrocompetent and chemically competent bacteria upon their respective LV plates with ampicillin and incubated them for 16 to 20 hours at 37 Celsius degrees, the following results were obtained. In Figure 1, the left image shows electrocompetent cells that could have been transformed with the plasmid, totally represented by white-colored colonies. The right image shows the UFC counting made through paint to obtain transformation efficiency regarding electrocompetent cells. In Figure 2, the left image shows chemically competent cells that could have been transformed with the plasmid plus the gene of interest, totally represented by white-colored colonies. The right image shows the UFC counting made through paint to obtain transformation efficiency. Transformation efficiency of chemically competence and electrocompetence are shown in the following table.
Regarding chemical transformation, there's the need to take advantage of the cell membrane composition whose lipid molecules are negatively charged. Inside a cell membrane, there are adhesion areas that could allow the entrance of certain molecules such as DNA. However, negative charges from DNA phosphate groups are repelled by phosphate negative charges from lipids in the cell membrane. Thus, it is important to use a salt, in this case calcium chloride, to neutralize lipids negative charges so that DNA could be able to enter to the cell by its membrane. Also, there's the need to decrease temperature to stabilize the cell membrane and then a heat shock by increasing temperature at 42 Celsius degrees so that the cell membrane permeability joined with its neutralized electric charge will allow DNA to enter to the cell. On the other hand, electroporation consists of applying an electric pulse by a short period of time, bringing about an alteration in cell's membrane, conductivity, and permeability. Nevertheless, this process requires a cell suspension that possesses a high resistance and low conductivity so that DNA insertion can be ensured and bacteria will not die. One important thing is to add DNA free of salts and check if bacteria have been desalinated. Otherwise, salts will conduct electricity leading to cells death. When comparing both techniques, we have the following differences and purity requirements for achieving an efficient transformation. According to Thermo Fisher Scientific Catalog, E. coli top 10 one-shot chemically competent cells must have a transformation efficiency of 1 by 10 elevated at 9 UFC per microgram plasmidic DNA. Then, when comparing the transformation efficiency of chemically competent cells gotten in the genetics engineering laboratory of 3.34 by 10 elevated at 4 UFC per microgram plasmidic DNA, it is clear that there was an efficient transformation process due to the following reasons that will be discussed later on. On the other hand, E. coli top 10 electrocompetent cells must have a transformation efficiency of more than 1 by 10^9 UFC per microgram plasmidic DNA. When comparing this value with the value obtained of 4.62 by 10 elevated at 4 UFC per microgram plasmidic DNA, it is clear that there was also an inefficient transformation process.
None of the cells prepared by the team can be used to transform neither a supercoiled plasmid nor to take ligations because there was no growth in any of the experiments done. For this, samples of other teams should be borrowed. The steps in which the team could have failed are unfrozen liquids that could have led to cell death, contamination with proteases or DNases of the members of the team hands. Maybe the cells were never competent because of incorrect measurement of the reactives, which led to the selective death of all cells. These mistakes can be avoided by not leaving the liquids that contain the cells out of the ice at any moment. Not taking the gloves off until the experiment is over can also be an alternative.
Also, incubation time probably was not the appropriate as it has to be 37 Celsius degrees for 1 hour. Regarding chemical transformation, a heat shock wasn't performed at an adequate time, which consists on increasing temperature at 42 Celsius degrees for 45 seconds, thus membrane permeability couldn't have been altered. Another factor that could have affected are that the cells weren't in log phase while the transformation process was done, and that is, we froze the samples before transforming them. Some could have died in the process. To sum up, chemical transformation and electroporation differ each other in the type of reactives used to introduce competence in cells. For instance, chemical transformation uses salts for neutralizing cells' membranes and a heat shock for modifying a cell's membrane. On the other hand, electroporation bases on giving an electric pulse for a short period of time that tends to alter conductivity and permeability of a cell's membrane. Remember that cells lacking of salts are needed for electroporation. That's why glycerol solution is applied for desalinating a cell suspension. Whereas, regarding chemical transformation, cells need to have a regulated concentration of salts in order to carry out the process. Finally, it is important to absolutely follow the protocol and sterilizing measures for avoiding low transformation efficiency values when compared with the commercial ones.